Hey everyone, welcome back to genetics. So far in chapter 3, what we've covered for the first section was mitosis. Now this is where, stuff gets interesting for genetics. And it's basically meiosis. Meiosis is the most important process for us to generate the gametes. And then these gametes can explain how the law of independent assortment works and the law of segregation works as well, okay? So what is meiosis? I'm pretty sure most of y'all sort of are familiar with meiosis, but this is a process by which you generate genetically different and these will be haploid gametes most of the time. Haploid gametes. So basically this is what it is. You're generating haploid gametes. And with this process, you end up having two stages. You have meiosis 1 and you have meiosis 2. And essentially what their phases are, they're basically the exact same phases as mitosis. You would have prophase, you would have prometaphase, metaphase, anaphase, telophase, and cytokinesis. But for genetics, especially for transmission genetics, we really don't care about the nitty-gritty stuff about exactly how prophase works, exactly how telophase works. We don't really care about that. What we care about is how we're getting different proportions of different gametes. There are some important things to know for meiosis 1. Meiosis 1, this is where you have homologous chromosomes separate. Another thing that happens during meiosis 1 is something called crossing over. And this happens in prophase 1. So essentially what crossing over is, we're going to talk about it a little bit more in detail. But it's basically when homologous chromosomes, they join together and they can exchange different alleles from one homologous chromosome to the other. But for now, this process isn't really that important. It's going to come into importance when we're talking about gene linkage, but for now, it's not that really important. And then for meiosis 2, this is essentially where sister chromatids separate. And that's basically most of the stuff that you actually need to know for meiosis. And I drew out a little bit of a flowchart, basically how this happens. Let's say you have a genotype. Let's say you have a genotype that is big A, little a, big B, little b. So I've mentioned something called a homologous chromosome and a sister chromatid in previous videos as well. but basically, what are they? And whenever you have a definition, you can get kind of confusing because you're not really visualizing what they are. Homologous chromosomes are basically chromosomes that come from different parental origins, but they have the exact same genetic information. So basically, they have the same genes, but they can have different alleles. And what's important is that they come from different parents. So for example, a homologous chromosome in this situation would be this blue big B chromosome and this pink little b chromosome. So that's basically what a homologous chromosome is. And same thing for the pink big A and the blue little a chromosome as well. Another thing that you might have, like a homework problem, is distinguishing which are homologous chromosomes and which aren't. Sometimes you're not going to be given an easy peasy like a gene annotation on your chromosome. The way how you determine is every homologous chromosome has a different length, and that's how you can determine if a chromosome is homologous to another chromosome. Okay? Cool. So that's basically what homologous chromosomes are. And then during S phase, what's going to happen is you would have this genotype. You're going to double everything. You're going to copy all of them up. And a sister chromatid is basically the exact same copy of a chromosome from that specific parental origin. So basically, for example, a sister chromatid will be these two little A's, or these two big A's that come from mom. So those will be sister chromatids. Okay? So basically, that's what they are. I will say take a minute to try to remember what they are, because everything that you're going to do in genetics from now on, you're going to have to think about how gametes are being separated out in meiosis. And if you're not really familiar which one is a homologous chromosome and which one is a sister chromatid, you're going to have a really hard time with the rest of the process, okay? And if you have a question about why did I draw them separated out like this, how I have some chromosomes that are really far apart in like different orientations, that is extremely important, because whenever you have the genotype, your chromosomes are never going to be aligned, and they're only going to be aligned during the phases of separation, which is metaphase, anaphase, and prophase as well, where the homologous chromosomes are going to be aligned, stuff like that. Okay, so during meiosis 1, like I said, homologous chromosomes separate. So remember, homologous chromosomes are chromosomes that come from different parental origins and they contain the same genetic information. So basically what's going to happen here is that your big A's are going to be separated away from your little a's. So it'll be something like this. And then the other cell can get the little a and the big B. So hopefully that makes sense. And then next step, you're gonna generate these haploid gametes, which are just N. Basically what's happening here is you're gonna have your sister chromatid separate. So basically the big A from mom are gonna separate it into two different cells, where one cell is gonna have big A and then the other cell is gonna have big A too. And that's basically it for meiosis. And then I put up a little bit of a practice problem for you guys. See if you can really get it. But like how do we actually get Mendel's laws through meiosis? In other words, like the law of independent assortment states that if you have more than two genes, their alleles, their gametes will separate such that every single possible combination of alleles will occur at equal frequency. Same thing for a law of segregation. If you have just a single gene, you would have an equal proportion of both of those alleles occurring in your gametes. So how is that happening for our practice problem? So what we can do is we can go back over here, and the law of segregation is probably the easiest one to describe. What we can do is we can only look at the big A and the little a alleles, that only the A gene, and looking at how the A gene is being separated, what we can tell is that during anaphase 1, where these homologous chromosomes separate, these homologous chromosomes basically have you ending up with an equal proportion of your gametes containing both the, so this would be 50% chance to get big A, and this would be 50% chance to get little a. So basically, anaphase 1 is responsible for you to get the law of segregation. Basically, whenever you separate your homologous chromosomes, so your A genes, your A alleles from mom are on the top of the top cell. The bottom cell has the dad's little a alleles. This is basically the stage where you separate your alleles out from one another. So that was basically the easiest one to solve. So hopefully that kind of made sense. Another, for the law of independent assortment, this is where you have two different genes, two or more genes, and every single possible combination of alleles occurs at equal frequencies. So looking at our model right now, what we have here is that, let's say we have an individual that is big A, big B, little b. We would have a total possible combination of 4 different alleles from this genotype, or allelic combinations. But what we're getting here is something like big A with little b, and little a and big B. So how do we get the other possible combination of alleles, which are big A, big B, and little a and little b? So what's happening with them? So the way how you can think about this one for the law of independent assortment is we come back to the to this figure right here during metaphase 1. What we can do is remember that cells don't actually have directionality, and the chromosomes kind of line up randomly across the metaphase plate. There really isn't a set way how these homologous chromosomes should line up. So basically what I mean is that these little a sister chromatids can actually line up to the left, and you will get a combination that is something like this. So basically what you can have here is you get a combination that can, I should actually just say this, or, so basically you get a combination like this where you have a different set of homologous chromosomes line up differently on the metaphase plate. And then from this, what you can do is you can separate these out into 2 different cells. And for time's sake, we can just write down little a, little b over here. And then for this one, we can write it down as big A, big B, big B. And then remember, during this stage of meiosis, we're going to separate our sister chromatids. So basically, we would have one cell with little a, little b, little a, little b. And this one is big A, big B, big A, big B. And just remember that during gamete formation, you have millions of rounds of meiosis occurring every single time. So basically what's happening here is that yes, you are getting little a, little b here, and big A, big B here. But at the same time, these gametes are also being made, so they combine together to get an equal frequency of, this will be 25%, this will be 25%, these will be also 25%. And then that is it. That is basically meiosis for y'all. So hopefully that helped you out, and that's it.